It's really great to be here to help kick off this new institute. I'm excited to see all my Hopkins colleagues who are going to participate or who are excited about it themselves, and our guest speakers as well. I was kind of enjoying listening to Mike's talk, mentioning personalization. I spent a long time at Google, and actually when I was part of the search team there, we worked on launching personalized search. So all of the things you described were the things I heard as we were launching that. But since then, I've really turned to a new problem, which I think is much more challenging, but related, which is personalized genomics. So what I'm going to be talking about today is personalized genomics and a project we've been working on to try to address some of the challenges there. The overall goal of personalized genomics is to figure out if we can understand the effects of individual sequence variation in the human genome. So basically, if somebody comes into a clinic and they get their whole genome sequenced, we'd like to be able to tell them what diseases they're at risk for, what the effects will be on the different tissues of their body, et cetera. So to be a little bit more specific and to introduce some terminology, what we're really trying to do is predict the impact of DNA sequence variants. So a genetic variant is simply a difference in the DNA sequence in a specific individual compared to the normal human genome or compared to other people. So I'll be using this term a lot, genetic variant. It's simply a change in the sequence compared to normal. And our goal is to look at one of these variants and predict what will the effect of this change in sequence be. Will there be changes in cell state in different tissues of the body? Will there be an increase in disease risk, et cetera? So we'd like to be able to make these predictions. So as a little bit of background, you probably see in the news a lot of studies coming out saying, oh, we found a genetic risk for schizophrenia, a new gene is discovered that's relevant to a particular disease. Most of these that you see in the news and in literature are based on what's called genome-wide association studies. In this type of analysis, a set of individuals are collected, some who have a disease of interest and some who are healthy controls, and we look at their genetic profiles, and we go through basically variant by variant, and we simply test for an association, a simple correlation analysis, variant by variant, to give us an odds ratio and an associated p-value that simply tells us whether a specific change in DNA sequence that we see across the population confers a greater risk for a disease we're interested in studying. So this is called a genome-wide association study. However, this type of analysis, the simple correlation analysis, is really only amenable to testing variants that you see many times, right? These are what we call common variants. And there's lots of common variants in the population, and they are, as you see in the news, in fact, often associated with changes in disease risk. But this type of simple correlation analysis really only applies to common variants. And you probably don't need someone like me to run a genome-wide association study. So the problem that I'm interested in is much more challenging than this. So it turns out that many of the interesting changes in your genome and in any of our genomes are actually unique to you or just a few other people. So if I were to go look at a genome-wide association study that's been run, many of the variants in your genome simply were not tested and cannot be tested through simple correlation analysis like that. So how do we predict the consequences of what we call rare variants when association methods simply can't be applied to them? So this is where my interest comes in. So we really have to generalize from other variants we've seen before. So this is kind of a perfect opportunity for machine learning, which is my specialty. So to motivate this a little bit further and to give you a sense for how hard the problem is, each of you are walking around with something like 50,000 rare variants in your genome. So these are variants that are seen in less than 1% of the human population. You have about 50,000 of these in your genome. So if you were to walk into the clinic and get your whole genome sequenced, your doctor is then looking at 50,000 variants that honestly we have very little idea what they do. So it's extremely challenging to identify their effects. Most of these variants are not inside of protein coding genes. So if you took biology as a student or even a high school student, what you learned about the genetic code and coding for proteins simply doesn't apply to 98% of these. So you're looking at tens of thousands of variants that your clinician has basically absolutely no idea how to interpret. So the realization of clinical personal genomics is limited by this problem of rare variants and these very challenging interpretations. But it's really important because if we ignore these, obviously we're missing potential major effects. So what we've been working with, so in addition to an explosion in whole genome sequencing and our ability to sequence your entire DNA sequence, There's also an explosion in other types of data, based often on sequencing, but that give us a very different view of what's going on in your body. So we call this multi-omic data, and we're gathering data that tells us things about protein levels in different tissues of your body, that tells us things about the structure, how your DNA actually folds up in 3 dimensions in the cell, about protein levels, RNA levels, all sorts of things like this. They give us a much more dynamic and multifaceted view of what's actually going on in your specific cells in your body. And so we're using this data and actually a very simple insight that in addition to your DNA sequence, we can look at your molecular profile and see where you are extreme relative to the population in ways that might correspond to your genetic changes. So we look for individuals who display very extreme molecular changes in their cells in combination with looking at their DNA sequence. So the goal of our system is really going to see how far we can get towards this personal genome interpretation. We would like to take every variant in an individual genome sequence, and for every one of those variants, provide a probability that that variant is actually doing something to that person. It turns out that many of these 50,000 variants that you're walking around with actually do little to nothing and are not going to cause you health problems, and they may not even do anything noticeable to your cells. We'd like to identify the ones that do. So we want to estimate a probability that any given variant is having some impact in a given person. So luckily, given that I want a machine learning framework, there are lots of features we can use. So the human genome has been studied in depth and annotated with all sorts of informative features that we can dump into a system like this, things that tell us evolutionary conservation of different pieces of the genome, data that tells us where important proteins bind to the DNA itself and whether a variant is inside of one of those binding sites. I won't go through all the biology behind this, but there is just an explosion in this type of information. Now, this is not personal level information. This is annotation of the human genome as a whole, collected from background sets of people that are not your patient that's in your clinic, but it's still telling us a lot that we know about every different position in the human genome. And we can use these features to try to make predictions for variants we've never seen before. And people have been working on this. So there are recent methods for scoring variants that are based on whole genome sequencing and this sort of background data alone. So, you can think of a simple model like logistic regression or a support vector machine, things like that, that simply take annotation of this particular region of the genome where you see a variant and simply try to predict whether or not it's going to have an impact. Now, most of these, however, rely on supervised learning, and this is really a problem because it turns out that there is not a nice, big, unbiased source of labels that we can use for a supervised learning problem in this setting. I told you we don't really know what most of the variants in the human genome do. That is true. So getting a training set of variants that we know do something and a set of variants that we know don't do something is either going to be very, very small or very, very biased towards things we already understand. So that has been really problematic. But there are a number of methods that attempt to do this in a variety of ways. I'm not going to directly address this problem. In fact, we're going to use some of their scores as features in our model. So we're not going to try to improve on the supervised learning version. We're going to try to take it a step further and see what else we can do. So our insight, again, getting back to this explosion in sequencing data that gives us a molecular profile of our individual, is we can go beyond simply taking a genetic variant and from a whole genome sequence, combining it with this background information and estimating the probability of impact. What we're going to add to that is getting molecular data in our actual same individual, our patient. So we're going to get this molecular measurement, and we're going to go in and look at that person and see, are they very extreme in the expression or protein levels of a gene that might correspond to that genetic change. And we're going to see how much further we can get by doing this. So we developed a method called River, which is an integrated model that allows us to combine both background information based on whole genome sequencing of our individual and known properties of the human genome with expression data from that same person and whether or not they display very extreme expression levels. Now, the important thing that we're trying to predict is this hidden, latent variable of whether the variant that we're looking at has a functional impact or not. So this is this is really what we care about, but it is going to be entirely unobserved. So our method is completely unsupervised in that sense. So no labeled data is needed for training at all. You don't need to know a set of variants that are bad and a set that don't matter. But at the end of it, at the end of training, our framework is still able to provide a principled estimate of that posterior that we wanted, the probability that this given variant has a functional impact, by combining both their genome sequencing and their gene expression or molecular data from the same person. But it's trained in an entirely unsupervised way. So, the work that my lab does, basically we try to think about the diverse sorts of data we can use, the specific relationships that might be encoded in this type of data, and write down a simple probabilistic model that tries to put it all together. So that's exactly what we did here. It's a very simple probabilistic model. The top half of it, if it were given observed data, is really just logistic regression. The bottom half is really just naive Bayes. We have this latent variable. We don't require supervised data, but the whole thing can be trained very efficiently just using expectation maximization to identify the parameters of each of these distributions. It encodes what we think is a very simple process, that a functional variant should have some effect on the cell, and that information about the genome should be informative about whether a variant is in fact functional. It just encodes all of that, and we're able to train it very efficiently. So we took this model River and we applied it to a recent data set that is one of the largest of its type. It is from the GTEx project, which I've been part of for a number of years, and it provides for us gene expression data from about 500 different individuals across multiple tissues of the body. So they're collecting gene expression data from around 44 different tissues for about 500 people. Of those 500 people, we actually also have whole genome sequencing for a subset, and that's actually growing. So this is a really rich data set that gives us a really strong picture of what's happening molecularly across the body of these individuals, including in brain, lung, liver, et cetera, in addition to their whole genome sequence information. So we applied River to this data, but as I told you, labeled data is hard to come by. So even after we trained this, just like it's hard to train a supervised model, it's actually hard to evaluate our model. So because this labeled data is not available in large quantities, we had to come up with kind of a roundabout solution. So our idea was to actually take, for variants that we define as rare at 1% minor allele frequency, in this data there are a number of genetic variants that we see exactly twice. So we have 2 individuals that share the same variant. So what we did was remove all those N of 2 cases from our data for training. So we trained River on GTEx without any of those N of 2 variants. Then we took one of the people that had a shared, this N of 2 shared variant, and we used the trained River model to make a prediction. We computed the posterior that variant had a functional effect given that person's whole genome information and their gene expression molecular information. So we made a prediction based on one person, and then we evaluated it in our completely held out individual that shares that genetic variant, but whose molecular data we've never seen, and we actually evaluated against whether that person displays an extreme change in their molecular data as well. So it's a noisy label, but it's a label that we've never used before and actually allows us to evaluate our model. So we did run River and we evaluated it in this fashion. And what we see is that this use of molecular data in addition to the genome information gives us a significant boost in performance. So River is in blue, it's the higher ROC curve there, and the model that is like the other supervised models that I've described, uses genomic annotations alone, performs much worse than River in this case. Now you can see that the AUCs are not super high, right? We're getting something around 0.6, 0.65. This is a very challenging problem. As I described to you, we really don't know how to prioritize these variants yet. Our use of molecular data is certainly giving us a big boost, and you can kind of see over, in the left-hand side, it's giving us a boost where we're able to identify a number of true positives before we really start incurring much of a false positive rate at all. So it's identifying some very high confidence variants that our system is able to pick up before it starts making mistakes, but the problem remains extremely hard. I will note that one of my students, Ben Strober, has recently been working on an extension to this that really only looks at a subset of variants. It's a type of variant that we know a little bit more about. And when we apply this system in that case, we still get a big boost. And you can see that in this limited case where we're looking at for variants that can affect a process known as alternative splicing, overall the performance is much better. But this is limited to a small subset of variants. But we're quite excited about this progress. So we've applied this system to try to really understand, could this be taken and used in a clinical context? So what we did was collect a set of known pathogenic variants from a database called ClinVar. Now again, you might think maybe we should have used that for training. It's very small, and it is very biased. It's actually biased much more towards variants that exist inside of known genes that have known effects and things like that. So I would not want to personally use it for training, but we still wanted to see whether our system, River, could prioritize known pathogenic variants in our individuals above their sort of background of their other 50,000 variants that they're walking around with. So what we did was then evaluate whether the molecular gene expression data alone was informative about which variants were pathogenic. We looked at whether genomic annotations alone from whole genome sequencing alone was informative. And finally, we applied our model River. And you can see that each of the two signals that we integrate, of course, has some information. You do get some prioritization of pathogenic variants from only molecular data. You do get it from only whole genome data. But by using it together in this combined model, we see a much greater prioritization of these variants, and again, a subset of them that River is very, very highly confident about. So it's able to pick out a few of these that have clear molecular effects and prioritize them very strongly against this background of this huge number of rare variants. And specifically, if you look at all the regulatory pathogenic variants from ClinVar, all of them ended up being in the top 0.1%. According to River posterior. So River's really highly prioritizing these known pathogenic variants. So what would this look like in clinic? So Ben has also recently started applying this to a set of undiagnosed genetic disease cases. So when these patients come in, and they have, in this data set, approximately 70,000 rare variants each that we're looking at. That clinically is not manageable, right? That is not at all manageable. Even if you restrict to variants that are pretty close to genes that might likely be functional, you're looking at something like 30,000 variants. Again, not manageable in a clinical context. But when we start using this idea of molecular data along with it, from these patients, when we're looking at changes in total expression or this alternative splicing process that Ben has started studying, we're now able to get it down to what we think are the most likely functional variants from these, where we have like 4 or 5 for a doctor or clinician to look at to try to give our patients some input as to what might be going on in the disease context. So we're really excited about the applications of this system now. I told you about some of my interest in a particular project we're working on, but as we're talking about the launch of this institute, I definitely want to mention that there's a large group of people working on genomics here at Hopkins. I took this slide from Mike Schatz at some point, really ranging all the way from devices that can help us improve sequencing all the way up to things like machine learning and actually trying to apply this type of thing in clinical context. And this is just really a subset of the individuals. I didn't have room or time to write down every single person here at Hopkins who works on these ideas, but there's a lot going on. So really what I want you to walk away with is thinking that, I showed you this idea of using an integrative approach for personal genomics. If you'd like to play with it, there's a bioconductor package available. But what I really want you to take away from it is that there's this deluge of data, genomic data, molecular data, that in theory should allow us to identify some exciting and novel effects of genetic variation and move us towards personal genomics. But my perspective is that if we use really careful and tailored methods to leverage this data, not just in sort of a black box machine learning approach, but really think about the relationships in the data and what it's telling us, I think it can take us a lot further. So thank you for your time.